topic of clinical trials, clinical trial readiness, and the challenges we're facing with that. It's really so inspiring for me the last 2 days, and especially, hearing patient stories. This is what drives our research and our daily work also in the clinic. So it's really very powerful, and I would like to congratulate you all for your leadership, for your involvement, and also the CMTRF, of course, for bringing all this together. So I will start with this. So I was asked to discuss why is it important to visit the CMT center. So here you see a patient's testimony that I found on the MDA website. So throughout the day, I met with numerous experts, all with the intent of helping me and better educating me regarding CMT and possible treatments or things I could do to help myself. So that basically encapsulates the experience that patients have in CMT centers. And probably some of you had this thought that for the first time you see a physician who knows more about CMT than yourself. And unfortunately, many times you have seen physicians who know much less about CMT than yourself. So I will just start with a reminder of what we're talking about and the Charcot-Marie-Tooth neuropathies as a whole. This is a disease that affects 1 in 2,500 individuals worldwide and has a very variable presentation with onset from early childhood to late adulthood and old age. It presents with weakness that is slowly progressive and loss of sensation, impairment of balance, and it is very slowly progressive for most patients. The important challenge here is that it's a very heterogeneous disease. So you see here the map of the chromosomes, of the human chromosomes, and the genes involved in CMT are all over, including the X and all other chromosomes, and there are more than 100, and probably we still haven't found them all. So that creates a big heterogeneity in the disease. Having all these genes means we have a lot of different causes of the disease. Even we heard before the final presentation may be similar. There are very different pathways that lead to this presentation. So we have diverse cellular pathomechanisms and molecular mechanisms. And as you all probably know, we classify CMT according to how it presents, whether it affects mainly the axons or the myelin. So axonal CMT, CMT2, it's autosomal dominant, the inheritance, and it is caused by mutations in a big number of genes that are very important for the function of the neuron. So you see here a neuron and the axon that goes all the way to the muscle, for example. And so many of these genes have to do with the transport of material along this axon, which can be 1 meter long in our body, or with regulation of producing protein and vesicle recycling and excitability. So they start damaging the axon as a primary event. And then we have the demyelinating CMTs, CMT1 and CMT4. CMT1 types are dominantly inherited and 4 are recessively inherited, but they all affect genes that are important in the cells that form the myelin insulating sheath, the Schwann cells. And some of these are some of the most common ones are myelin proteins. So there is PMP22 and connexin 32 and myelin protein 0 and some other ones. And some other ones have to do with the regulation of this cell development and integrity. So a lot of different mechanisms, but in the demyelinating neuropathies, gradually you also get the damage of the axon, and that's a secondary event, but that leads to the symptoms that we all know. So let's deal with the first challenge. And the first challenge in developing treatments for CMT is the rarity. So they're rare and slowly progressive. And you see here that, for example, the most common type, CMT1A, affects 1 in 5,000 people. This is the estimated prevalence, maybe under-recognized in some places. The second most common, the 1X, affects 1 in 25,000 people. And then from there and further down, they become even more rare. So CMT2A and 1B affect 1 in 36,000 of the population. The short neuropathy, for which we now have a clinical trial ongoing, affects 1 in 100,000. And then some of the most rare ones, like 4C, affects less than 2% of all patients, and 1B less than 1%. And this has led to problems. For example, it has been difficult to recruit more than 20 patients in the CMT4J natural history study that was going on for a while, a few years ago. It has been difficult to find more than 50 patients in the CMT4B study, because there is a treatment coming up, but we cannot collect enough patients. And it has been even challenging to recruit patients in the short clinical trial that is going on. 100 patients were needed. So here you can see that patient numbers are important, and being so rare, that creates a big challenge. Now, the progression is another issue. You heard a lot about it yesterday, about the fact that, most CMTs, and definitely the common ones, progress very slowly. And in the last 20 years, we used tools, evaluation tools, to see the progression that were not very sensitive. So the CMT examination score and the neuropathy score, and they did not really show a significant change over time. The example here is the ascorbic acid trial from the European side. And you can see on the top diagram that the score, which the more numbers you have, the more severe the disease is, doesn't really change over a period of 24 months, which is, a reasonable time for a clinical trial. You cannot run a clinical trial for years. And below you see the change in the score, which is close to 0 over the same period. The American ascorbic acid trial was even more concerning, because here not only patients didn't change, but actually some of the placebo-treated patients showed improvement. The placebo group was different from the placebo group in the European trial. It was different from the natural history, from what we expected from previous studies, and it was better than the treatment group. So all this led to the realization that we really have to work with patients, and we have to develop more sensitive outcome measures. We need more sensitive ways to measure the worsening of the disease and possible response to treatments. So at the same time, we have, of course, and you heard about it all day yesterday and today, the challenge of the long path towards treatments. And this path starts from discovery of the disease, understanding the cause of the disease. Some of this work is done in the dish, in cultured cells, in the lab. Then we move on to the small animals to model the disease. For neuropathies, it's important to have this, because that's the only way you can see the nerve and how the nerve is being damaged. And then we need to move to bigger animals to prove safety, and to prove that we can scale up the treatments to a bigger organism before we go to humans. And then we have the long path of clinical testing until we reach the desired approval of a treatment. So it's a long path. And where do we stand today? So drug treatments, the small molecules that you heard before, are a little bit more advanced. So we have a few clinical trials going on. I may be missing something here, but we have the PXT trial ongoing and the short neuropathy trial. So they're already in testing in humans. And for gene therapy trials, we're much more behind. So we reach the primates, and some of them, hopefully, like the DTX technology, will move to the clinic next year. And there is one for a very rare type, the CMTS, that is also in clinical testing, but that's it. And we do not have any approved, FDA-approved treatments. So this is the challenge we have to face. So how do we move forward? So it is very important, as I already mentioned, to develop more sensitive clinical evaluation tools. We have to be able to see that there is a deterioration of the disease within a shorter period of time, within a period that a typical clinical trial runs, which is not more than 2 years. And if you talk to our company stakeholders, they would like it to be 18 months or 12 months because it is very expensive to run a trial for a long time. And the other important aspect is to have biomarkers. So biomarker means to have a lab test or an imaging test that shows that there is a worsening of the disease. And hopefully it will show that even before the symptoms get worse, even before we can measure it in the clinic when we examine the patient. Sometimes the biomarkers will also show the target engagement. So they will show that what we're going to treat actually is being treated. So that means we need natural history studies. So I'm coming to the natural history studies and what they mean. So natural history studies will allow us to test more sensitive evaluation tools in the clinic. They also enable the development of biomarkers such as imaging. You heard some of this yesterday. So there is MRI to measure the fat fraction in the muscle. There is also microscopy to image the sensors of the skin. And ultrasound is being also developed. There are some very good data. It hasn't been until now, but the technology is improving and may become also very useful and more accessible biomarker. We also have blood biomarkers. Many of them have been investigated and they can be very useful. And we have skin biopsy, especially for CMT1A patients, because there you can see the reduction in the level of the excessive PMP22 that is in the cell. You can even see nerves in the skin biopsy, so that may be also a useful biomarker. So let's focus first on the clinical outcome measures. I already talked about the traditional ones, the neuropathy score and the examination score. They are very useful, but they're not sensitive enough. So what is being developed now is the CMT functional outcome measure. It's called CMT-FOAM. This is actually something that is being developed for adult patients. It was initially developed for infants and children. So there is a CMT infant scale and CMT PEDS, and now we have CMT-FOAM. This is a much more detailed and composite evaluation measure where we look at various aspects of function, including the hand function. There is a whole peg test, there is dexterity test, there is gait test, 10-meter test, get up and go. There is a lot of balance test. So it's a much more comprehensive evaluation. This has already been validated for CMT1A, and it correlates with the traditional tools, and it's much more sensitive. It doesn't have a ceiling effect. So it is going to be the main evaluation tool in future trials, for sure. In addition to this, we have patient-reported outcomes. They're equally important. So we have quality of life measures, the CMT health index, the overall neuropathy limitation score, and so on. And so this will be part of a clinical trial. We heard yesterday from Dr. Eichinger about the wearable sensors. This is another technology that is being tested. They can measure balance, they can measure mobility over extended period of time. And so they can be very useful tools in the future. The MRI has proven to be the most reliable and sensitive biomarker to date. So what it does is it measures the fat replacement in the muscle. So when the disease progresses and the muscles become denervated, they start accumulating fat tissue in the place of the healthy muscle tissue, and you can quantify that by MRI. The good thing about the MRI is that you can look at different places depending on the patient's stage of the disease. So if someone has a milder CMT type, you can look at the calf because that's where the fat accumulates and less in the thigh. If someone is more severe and the calf is already very atrophic, you can move up to the thigh and you can quantify the fat replacement in the thigh muscles. So it's something we can tailor to the stage of the disease. It has been evaluated over time. You see here on the right, patients of different ages suffering from CMT1A, and you see the progressive increase in the fat amount in the muscle. And there is now good evidence that there is a linear progression from the age of 18 until the age of 50 in CMT1A patients. And it can show the progression earlier than the clinical evaluation tools. So it's definitely something that will be part of any future natural history study or treatment trial. You see here also the comparison with a healthy individual on the top, and below is a patient with CMT. The other biomarker that is being evaluated is the confocal microscopy, in vivo microscopy. So essentially visualizing sensors in the skin of the fingertip. These are Meissner corpuscles. They are responsible for tactile sensation. And using this sophisticated microscope, you can actually image them. You can see how many they are under the skin. And you can compare a healthy individual on the left with a patient with CMT on the right, and you see the reduction in the number of these sensors. So that's another biomarker that is being evaluated and will probably be useful. And there are also the blood biomarkers. You heard about them yesterday. So starting with the neurofilament light, this is a molecule that is a part of the axons. And when the axons degenerate, it will be released and found elevated in the blood if you use very sensitive methods like the Simoa. And it has been shown to be elevated in patients with CMT, of various types of CMT. And it also correlates with the clinical severity. So a patient with a higher severity would have higher levels. We have tested this biomarker in the disease model, and we have seen, for example, in the model of CMT1X, that neurofilaments are also elevated in the model compared to a healthy mouse. And when we treat the mouse, the levels go down. We saw the same effect in the CMT1A model. So it is potentially a treatment responsive biomarker. The only problem is that if you look at patients over time, they don't necessarily progress. So the levels of the neurofilaments do not necessarily increase over time, because it has to do with how active the damage is in the nerve. And there is a steady state of this damage, probably in patients. But maybe they would be useful to show a response in the short term. So when you treat a patient, maybe you see a reduction of the levels within a few months before we see others. So they're still very useful. There are many more biomarkers that were evaluated in CMT patients, like the neural cell adhesion molecule and the growth differentiation factor. So they were found elevated in CMT patients. They also correlate with the severity. There is TMPRS55. This is a protein that is released by Schwann cells. So this is the only one actually that is a Schwann cell marker. And that was also found elevated and may be useful also for treatment trials. And some of these biomarkers we also tested in the disease models when we treated them, and they show a response. So both the NCAM1 and the growth response. So they may be potentially useful. We need to keep them in mind. We need to include them in our studies, and any natural history study will include a blood draw, and some of these can be used, to just see how things develop. Or in the future, we may have more blood biomarkers, and we can go back to the blood samples and check them again. So this is very valuable material if we have blood at certain intervals, for example, every 6 months in the natural history studies. So where is all this being done? So these are the CMT centers that drive most of this research. I put them here on the map. So there are several centers in the United States. This is the Inherited Neuropathy Consortium Centers of Excellence. They're funded by the Rare Disease of NIH. Unfortunately, the funding is going out next year, so that's a challenge the CMT community will have to face. And they're spread all over the country. They do all this biomarker development. They do all the natural history studies. And of course, they provide care. And there are also 2 centers in Europe, London and Milan, and 1 in Australia. In addition to this, the CMT Association also supports additional centers of excellence that they also provide multidisciplinary care. Some of them are the same as the INC centers, and they also participate in care. And you see they're more spread. You can find them in more places in the US and worldwide. There is one also in Belgium. So these centers are very important for conducting all the research that we need to reach the clinical trial stage and then to conduct the clinical trials. So what should the patient expect when visiting one of these centers? So of course, you expect to see an expert, a neuromuscular specialist, a CMT specialist. There is the electrophysiological testing and the nerve conduction studies that are very important. And I heard yesterday from patients that, when they had the studies in any type of hospital, they tend to be more painful. If you are in a specialized center, they know the disease, they know how much to stimulate the nerve, and the experience is much better. And then from there on, you see a lot of other therapies that are very important for the care. So physiotherapy is crucial, of course, orthopedics, orthotics, and then depending on the disabilities, depending on the age, you may see also social services, psychologists, occupational therapists, and so on. So it's a multidisciplinary team that is there to support patients and provide supportive treatments until we have therapeutics. And even when we get the therapeutics, these are the people that are going to monitor the patients that are being treated and all the outcomes. So it's very essential. And so patients usually will get all this when they visit the CMT center. They will get all the demographics, the family history, the examination, sometimes skin biopsy. Nerve biopsy is not done anymore because now we diagnose CMTs with DNA testing. And of course, a lot of supportive care that I already described. So that's already a very important reason to visit the CMT center. But there are more reasons. So first of all is to receive this multidisciplinary care. And even if it's not done very frequently, I think it's crucial for making a treatment plan for the patient, for communicating to the local neurologist, communicating to the local physiotherapists, because not all physiotherapists have expertise with CMT. Not all orthopedic surgeons know about CMT and know what is the best procedure if needed. So that's very important. Also, it is very important because patients will receive information about their disease, about the research developments, the latest results. They will be informed about any ongoing trials, about any forthcoming trials. They can participate in patient registries. I will come back to the registries and why they're important. They can participate in natural history studies because these are the centers where they run the natural history studies. And they can later on enroll in the treatment trials. Treatment trials are going to take place in these centers. They're not going to be available in every hospital around the country. And also there are cases that are undiagnosed, even after all these evaluations and even after this DNA testing, we still don't reach a diagnosis. And that's a case where it's very important to visit the CMT center because they communicate with each other. Either here or in Europe, there is an interaction. Difficult cases are discussed between the experts. And so you can benefit from worldwide expertise and experience if there is a case that is undiagnosed. So that's also very important. So natural history studies are not an intervention. So we don't really treat patients, but we collect and I have the 2-minute left sign, so I will go very quickly over this, but I already discussed it. So we develop all these outcome measures and the biomarkers. So that's the natural history studies. I bring here as example ACT-CMT, which is already running for CMT1A. It includes all of the outcome measures that I already discussed, and it's almost completed. So we hope to get the results of this study by the end of the year. So that would be very crucial, but we need to have similar studies for all other CMTs. So that's really an unmet need. Patient registries are also very important. Patient registries are not as detailed as natural history studies, but they include a minimal data set that is important for knowing how many patients there are out there with this disease, what is the severity, and especially for the rare forms, this is very important because many times companies will ask, so how many patients suffer from this disease? What is the market? Where can I find them? Are they accessible if we run a trial or if we have a treatment? So even if there is no natural history study, patient registries can be very useful. And so that's really important to do. So when we move to the drug development, we have to go, of course, through a very detailed preclinical development, which includes all the aspects that were already discussed, the safety, the biodistribution, the testing in different animals. And the purpose of all this is to show that there is at least hope for effectiveness in humans and that the risks are minimized. And so I had as example here the work we did for CMT4C, which is an autosomal recessive disease. It's a loss of function, so we use gene replacement to rescue the model of the disease. And you see here an example of the development. So first, design the treatment based on the cause of the disease, treated the mouse model. That's the proof of concept. And of course, we have many more steps that are still remaining and require more support and funding. We need a natural history study, which is not there, and we need also the large animal testing. So, and because Mary Kate is in the audience, I would like to congratulate her because she's part of the CMT4C task force, really leading the effort to develop the treatment for this disease, for the steps we still have in front of us, which is engaging the patients, supporting research, advocating for a natural history study. And they're already doing a lot of work, and I would really like to thank them for that. So we're planning a natural history study and a registry for this disease, the way I already described. I will go very briefly over now the next stage, which is the IND, the investigational new drug. You already heard about it. So this is the application to FDA with all the preclinical data. And then we move to clinical testing, hopefully for some of the diseases. And you all know the phases of the clinical trials. So phase 1 to 4. Phase 1 is mainly for the checking the safety. Phase 2 is to show an effectiveness in a small number of patients. Phase 3 is usually placebo controlled. And phase 4 is after the marketing of the treatment. The numbers here do not, of course, apply to CMTs, and which are very rare. So we need to plan and design trials that can be supported with lower numbers of patients, especially for some of the rare ones. And you heard some of this from Dr. Flanagan yesterday. And of course, there is a clinical trial design that includes all the expertise from the expert clinicians and you know, all the planning, who is eligible, which patients can participate, and so on. So there is detailed clinical trial protocol. We have someone needs to sponsor the trial. These are usually companies. These are very expensive studies to do. And that's why Novartis had to acquire DTX in order to run the clinical trial. Then we have the IRB, the Institutional Review Board, that approves the ethical aspects, makes sure that the benefit will be more than the risk for the participants. And of course, FDA has to approve the trial and then approve the drug if the results are positive. The informed consent is very important because you need to know everything about this treatment, what the trial will entail, what procedures are involved, and you need to know that all participation is voluntary and you can withdraw anytime. And then we have the conducting of the trial, which includes randomization, which improves the validity because we can just avoid the bias. The trials are blinded, but some of the treatments, that will be a challenge to know that you're not receiving the real treatment. And placebo, which is again an issue because for some of the diseases will be difficult, and that's why the trial design has to be tailored to the small number of patients and the challenges with that, but that's how clinical trials are done. I think I covered this. You know why it is important to participate in clinical trials and natural history studies, because this is the only way we can get to treatments and to help ourselves, but also other patients later on. So I will finish with this. We really need patients to take an active role because you need to join advocacy organizations. For some diseases, every patient counts because they're very rare, and there will be no development unless patients will really advocate for this. So you need to advocate to the industry, you need to advocate for funding. We also need to get everybody together. We heard a lot about collaboration in this space because we need every stakeholder. We need the clinicians, we need the basic researchers, we need industry in order to advance. We need the natural history studies, and you are crucial for them to be possible. And of course, we need registries, and registries is another important challenge because they are not unified. There are registries, but they are not communicating with each other, and we need to create disease-specific registries, so subtype-specific. So I think that the puzzle of the collaboration and making progress looks like that. We need the patient groups. This is the most important driving force. We need the clinical experts, the INC centers, so you have to visit the centers. We need the researchers because we still have gaps in the knowledge about the cause of some of the CMTs. We need industry. They are the ones who are going to deliver the drug at the end of the day. And we need the regulatory authorities. And so all of these pieces of the puzzle have to be really working together in order to succeed. And with this, I would like to thank you.